yeah, it's great. It's great. All right. People are wondering what's going on here. Hi, everyone. I'm Fraser Cain. I've been a space and astronomy journalist for over 20 years. I'm the publisher of Universe Today, and I like to bring you behind the scenes, talking to some of the people who are actually doing the science. This is going to be way out of my comfort zone today, but I think it's going to be a lot of fun. I'm talking to Dr. William Ratcliff. William, welcome to the show.
Thanks for having me. It's
All
great
right.
to be here.
The question I always ask people, who are you? What do you do?
So I'm an evolutionary biologist and an astrobiologist.
Oh,
So
you are an
I
astrobiologist. Whew. All right,
yeah,
that's
I
a
mean
relief.
that's
Okay.
it's a big tent. It's a big tent. So there's a lot of different ways to be an astrobiologist. And I am the variety that tries to understand the origin of complex life.
Yes. So I guess let's man, where do we start? Okay, so let's talk about the history of just
Yeah,
multicellular complex life.
yeah, yeah, yeah,
Um
yeah.
what do we think about how life went from single cellular organisms to multicellular organisms?
Great question. The first thing I want to sort of, get into people's heads is that it's not like it was a single thing. This didn't just happen once, and then we had this, renaissance of multicellular lineages. This is something which has happened many times in the history of life in different unicellular backgrounds. So we know of at least 50 different origins of multicellularity, each occurring independently. The very earliest ones happened in bacteria. So cyanobacteria, right around the Great Oxidation event. No shade to my geologist friends, but the event took a couple 100 million years. So not much of an event, but around 2 billion years ago, probably a few 100 million years earlier than that, cyanobacteria started to form filaments, and some of those, and they divided labor. They have real development. Some cells fix carbon through photosynthesis. Some cells fix nitrogen, which is it's easily destroyed by oxygen. So they have separate cells which close off and fix nitrogen, and then share those products with each other. Within the bacterial world, multicellularity has evolved a bunch of times, but it's never really gotten that complex. But it's kind of been around. Like, it's probably, honestly, my opinion is multicellular bacteria have probably been around since we've had bacteria, because it's really not that hard
Hmm.
to get a group of cells and then to select on the functional benefits of having a group of cells. Like, that process, that's what we do in the lab. That's pretty easy. But we don't really have a great fossil record pre 2.5 billion years ago, and the
Yep.
things that we do find are sketchy. You can usually figure out it's a cell because it looks like a cell, and you can 13C, analyze it and show that it passed carbon through a biological filter. But beyond that, you can't say much. Um,
So sorry.
yeah,
So if we look at
yeah,
all of the animal, but I mean all of the animals on
mhm,
Earth,
yeah.
they all stem from one single
That's right.
ancestor.
That's
And
right.
so,
One single transition. We have to be careful with differentiating between individual ancestors and
right.
populations.
But essentially one technology for going
Yep.
multicellular.
Basically, yes. Although the story is actually more complicated when you zoom in on the actual transition.
Right.
Um,
Okay.
Let me
Okay.
just,
But I
I'll get
guess
there, but maybe I can just paint a big picture
sure. Yeah. Yeah.
sweep
You
of,
know. Please
of
go ahead.
multicellularity
Yep.
on Earth. So bacteria did it and then basically between the Great Oxidation Event and the rise of oxygen in the Phanerozoic, which is about 1.5 billion years, there wasn't really that much happening. It was a fairly boring time. Um, what we did have within the eukaryotes, which evolved again themselves right around 2 billion years ago. So 2 billion years in the grand sweep of sort of Earth's history, that's kind of like there's a big pin in that point, right? Where origin of life, roughly 3.5 billion years ago. And then 2 billion years ago is you have cyanobacteria cranking out enough oxygen that the Earth becomes aerobic. Eukaryotes arise around that time. Just for our listeners, eukaryotes are these kinds of cells that resulted from a symbiosis between an archaea and a bacteria. And the archaea kind of was like presumably sort of the host cell, and the bacteria evolved into what are now our mitochondria. And so we're confident about this, and we can go into that whole story if you're
Sure.
interested. It's super cool. And then not much happened with multicellularity in that boring billion between 1 and 1 billion years ago. Then eukaryotes kind of took the ball and went nuts with it. So animals probably started around 800 million years ago. We're kind of relying on molecular clock data pre 650 million years. And that data is kind of sketchy because the assumptions that underlie it are just not true. But it's probably between 650 and 800 million. We can be confident about that. Fungi probably evolved around that time. Red algae were going actually about 1 billion years ago. So the seaweeds were starting off. 450 million years ago, plants evolved. We don't really know when fungi arose, but fungi, we do know that from simple fungal ancestors, complex mushroom forming ancestors, probably evolved 8 to 11 times independently
Wow.
using the same basic genetic toolkit that their ancestors had but weren't using for complex multicellularity. And then the green algae, they're crazy multicellular files. They've evolved multicellularity more than 25 times independently. One of those lineages became what are land plants. And we typically say, oh, they're land plants. But really, the hard thing there was colonizing air. All of these marine algae live underwater and they evolved underwater. And so
Right.
evolving to grow in out of the water, that's really hard. And so even today in modern land plants, you see the succession of things that start out like liverworts, where they're basically just like on the creek side, like laying fully on the soil because they can't transport, they have no way to transport water. So first you get a cuticle, then you get the ability to transport water, then you get wood, and you see this cool series of sort of evolutionary innovations in plants that allows them to compete for light and become 300 feet tall. And that's pretty much it. That's the grand sweep. And animals are the ones that we often really care about because animals, land plants, algae, and the macroalgae, so seaweeds, those are the 5 lineages that have really run away with multicellularity, evolved complex multicellular forms, evolved complicated cellular differentiation, division of labor, and development. And everything else, while there's many, many different origins, they've all remained relatively simple compared to those 5 lineages.
So with those 5 methods of using
Yep.
multicellular itty,
Mm-hmm.
multicellularity.
Yeah, yeah.
So is that sort of like a version of convergent evolution where you've got like bats and birds and insects all developing wings but using different parts of the body to do it?
Yeah, exactly. That's exactly right.
Right. Okay.
And it's and convergent evolution is kind of a nested term, so or concept. So first, for sure, we see convergent evolution of multicellularity in different lineages, and you can imagine that plants and animals are doing it for very different reasons. Like they're not really bats and birds are evolving flight for kind of the same reason. In fact, they're very similar. But land plants and animals, like they are evolving to form
Hmm.
groups and divide labor for fundamentally different reasons.
So multicellularity
Yeah.
has an evolutionary benefit,
Mm-hmm.
but it's not necessarily the same one.
That's right. That's right. There's a bunch of so exactly. And I think this gets at this big picture thing that like while we use the term multicellularity to describe these organisms which contain more than one cell and have integrated function, there are lots of different ways to be multicellular. There are lots of different reasons to be multicellular, and it's in some sense it's like it's a bit weird to group them all into the same group. Like they're really different. Like they do fit in the same basket for a good reason. They're multicellular, but they're not all of a kind. They are very different from one another. And you're familiar with the ones that are actually the most complicated. There's slime molds that do crazy things. There's ciliates, which form stalked fruiting bodies. Like there's all sorts of there are bacteria that live in caves that basically as soon as the water floods periodically, and as soon as the water touches them, they spew out like propagules from a multicellular fruiting body type thing.
Wow.
There's all sorts of weird ways to be, I mean, kind of alien, right? There's all sorts of weird ways to be multicellular, and there's a lot of different benefits
Right.
associated with multicellularity that either would favor the first origin of groups of cells
And so when
or
we
would,
think about
yeah, yeah,
99% of all life has gone extinct throughout
mhm,
history, if
yeah,
we know of 50 right now, it's got to be likely
mhm.
that that's orders of magnitude off the number of times that life has actually figured it out in ways we can't even imagine.
I think that's probably true. Certainly, I mean, in a sense, there are some robustness to the fact that clades diversify. And so even though the majority of animals or plants that ever had have existed are gone, we still know of those multicellular clades, right? And so there's kind of, unless the entire clade goes extinct, we would have some record of it in the modern day. And we definitely have fossil evidence of multicellular forms that bear no modern
Hmm.
counterparts and are very enigmatic.
And and
Um,
and like, are there examples then of fossil evidence where it might be showing some kind of
mhm,
multicellularity that
yeah.
we don't know what it is?
Exactly. Yeah. So
Huh.
there are these fossils from Namibia, right around the Great Oxidation Event, 2.1 billion years ago, which look like fried eggs. I mean, they're about the right. They're slightly smaller than a chicken egg, like in between a chicken and a quail egg, and they have this sort of frill and a dome. And we have no idea what they are. we're sure they're biological,
Yeah.
but no clue what the organism was doing. There's but they look pretty multicellular to me.
Now, you mentioned oxygen a bunch of times. So what role
Yeah.
does oxygen play in the struggle for life to survive and the reasons for it to gain multicellularity?
Yeah, no, it's a great
Multicellularity. I'll
question.
get this
It's
word by the end of this episode.
you can also use multicellular as an adjective. So
Yeah.
multicellular forms,
Multicellular.
multicellular.
Perfect. Perfect. Yeah. Yeah. Yeah.
um, so oxygen is one of those things that's like just game changing to Earth as a planet, right? It completely changes the way life interacts with the planet. It's a consequence of life. So it's one of those just geoengineering things at a broad scale that had to be pretty epic to watch, right? Because you're just precipitating massive amounts of iron out of the oceans. I mean, we're driving around in cars that are a result of the banded iron formations from 2 billion years ago when we were just dumping iron from the oceans, right? Like, it's cool. Uh, and it's highly toxic to cells, right? If you don't have the machinery to deal with oxygen, then it's going to kill you. So, that's actually fairly solvable. Organisms can make themselves oxygen proof, but oxygen is this fantastic electron acceptor. So it makes aerobic metabolism possible. Aerobic metabolism is far more efficient than fermentation. It's 16 times the ATP. So, and you can eat food sources that you couldn't eat if you were trying to ferment them. So, I work on yeast. Yeast, if you don't give them oxygen, they make you beer, right? They take sugar, they make
Yeah.
ethanol. But
Yeah. You don't want them to have oxygen.
you don't want them to have oxygen. But as everyone who drinks too much beer knows, there's a lot of calories left in that ethanol, right? You'll get a beer gut if you drink too much beer. And it's the ethanol that's providing a lot of that energy. And we get that energy because we can burn it with oxygen, right? If you give yeast oxygen, they'll burn the ethanol too, and they'll divide using that as an energy source. So there's all this non-fermentable carbon in the world. You need oxygen as a cofactor to eat. And if you can't, so in a sense, it's like food. It's like a key part of food. If you don't have it, then there's all the stuff you can't eat. So once oxygen becomes available, it stays at a very low concentration, on the order of, we don't really know, it's hard to pin it down, but it's probably on the order of 1% or less of what modern levels are for about 1.4 billion years. And then it quickly blasts up to near modern levels around 650 million years ago in the Phanerozoic. And so there's been this, so actually we work on oxygen and the role of oxygen, the evolution of size, which is a sort of fundamental thing. Like every multicellular organism has to get, it's getting bigger than its ancestor. Once you start adding multiple cells together, you get big. And a fundamental constraint of multicellularity is diffusion limitation. It's a cost to everything that forms a group initially, that you're just going to be now this big inert thing that if you want stuff from the outside, it's got to diffuse through multiple layers of cells. That's hard to do. It slows down a lot, especially if you're consuming it actively. And so there's been this idea for a long time that sort of oxygen is this accelerant on multicellularity, that the more you have, the more complex types of multicellularity you can have, the larger organisms you can support, because oxygen can diffuse further before you evolve a circulatory system. And so it's kind of this thing that, the more the better. And we've done some recent work that actually kind of challenges that notion. Rather than being a monotonic benefit, we've actually shown with experiments and with mathematical modeling that oxygen has this sort of interesting Nike swoosh relationship with organismal size, that when there's none of it around, well, then organisms don't care about it. They're not competing for it. They're not trying to use it. But as you begin to add oxygen to a system, then actually that becomes a valuable resource in its own right. And if there's a trade-off between being big, forming a big group, and being able to eat that oxygen efficiently, just due to diffusion, and there is this trade-off, then in fact, there can be reasons to get smaller, to more efficiently compete for this limiting oxygen. And then if you continue pumping oxygen to the system, well, then in fact, you can kind of have your cake and eat it too, because you can be big, but oxygen diffuses far enough within you that you don't have to be small. So then you kind of relieve that constraint. And there's this interesting thing that if you have some oxygen suppression of multicellular size, because the evolutionary incentives are to get smaller, is more gain to being smaller and eating the oxygen than to being bigger and getting whatever benefits of group size exist in your environment. You can actually, as you keep adding more and more oxygen, you can make that suppression stronger and stronger, because you're just amping up the benefits of being small. You're providing more and more metabolism you can get from oxygen until you pass a critical point, and then it becomes beneficial again to get big. And so we've shown this with some cool experiments in our lab, and it's a very robust result from first principles modeling, that you can vary the assumptions of the model dramatically and still recover this result.
Now you mentioned this idea of a boring period
Yeah, the
when
boring
oxygen,
billion.
the boring
That's what they
billion.
call it.
Yeah, when oxygen was present,
Yeah.
but you weren't seeing this arms race in size and multicellular life. So
Right.
why?
We don't know. And I don't know if we'll ever know. So one of the things that you have to accept if you go into my field is that it can be very difficult to have a strong confidence that we're able to explain the past. Some things we're very confident in. The broad sweep of the way in which life has unfolded, the pattern of inheritance and diversification. We're pretty confident in that. Like we are very confident that all living things that we've ever seen share a common ancestor. Like some of those results are easy. But some of these causal arguments, there's a lot of different variables, and differentiating between competing hypotheses can be very difficult. So, for example, in the case of Earth's history, we don't really know if the reason why we didn't have much happening in the world of multicellularity from 2 billion years up to about 650 million years ago, or even really 1 billion, that's when red algae started doing their thing, is that because oxygen was too suppressive? There was just, like it was too hard to get. If it had been anaerobic, maybe they would have been able to get big because they didn't care about oxygen, but you add enough oxygen to the system that's not a winning ecological strategy. Is it, the big confounding variable here is it something about the cell biology of the ancestors that existed at that time? I mean, there's really three major hypotheses. Is it something big about Earth's state? Is it something about the prerequisites that you need in terms of the state of the genetics of the ancestor? And I can talk more about that. That turns out to be very important. Mm-hmm. Or is it something about the types of ecology and the selection on organisms that drives multicellular innovation, right? And so all three of these things are changing a lot. And so it's very difficult to try to ascribe causation to any one of them.
But it feels like, if this oxygen was present and we've seen life utilize this many times, figure out mm-hmm. how to go multicellular many times in the past, that there was some rate limiting step in that period. And my instinct is mm-hmm. complexity, that there was some mm-hmm. there was some very mm-hmm. rare thing that life had to figure out to be able to make that next step during that period. Because when there's lots of oxygen present, it seems to do it mm-hmm. many times, and you can make it do it in the lab and so on and so forth.
Right. Although, interestingly enough, in the lab, the easiest thing for us to do is to evolve them without any oxygen. In fact, that's way better than giving them like 20% of modern oxygen. In our system, if we evolve them with like open tubes where they're open to the atmosphere and they're consuming oxygen as it diffuses in, even if we put them through 6,000 generations of strong selection to make bigger groups, they get 2-3 times bigger than their ancestor. If we do it with no oxygen, at that same time, they get 20,000 times bigger. And if we pump in tons of oxygen, well, those are harder experiments. So we haven't done them as long. But if we do that over a shorter period of time, they also evolve to be larger very quickly. And so you can really see that the constraint of sort of intermediate oxygen can be a very strong constraint on size. It's just the benefits of being big have to really outweigh the growth penalty of being big.
So let's talk a little bit about your research and your work with yeast.
Yeah. Mm-hmm.
So what is the experiment that you're running?
Yeah. So we are doing a long-term evolution experiment in the vein of Rich Lenski. I'm not sure if you're familiar with his work.
The longest running evolutionary experiment ever.
Yep. They're on 75,000 generations in their E. coli. He's been doing this for over 30 years. And in fact, he just passed over the reins of the experiment to Jeff Barrick at UT Austin, who was his former postdoc. And Rich is going to be retiring. And to me, it's a very foundational experiment. It's not just inspiring. It's something which has kind of enabled my career and our approach. I wouldn't have tried to do what we were doing if Rich's work hadn't come first. And so we were actually trying to do a similar version of what Rich has done. So Rich Lenski in this experiment, they're taking E. coli and they passage them every single day, where they undergo 100-fold exponential increase in numbers, then they drop it down 100-fold. And they just keep this exponential growth and passaging going over and over and over. And you can accumulate 6.6 generations a day. And now they're on generation 75,000. And you can watch all these cool things about how evolution works, because we have this, and you freeze them every 500 generations in a -80 deep freezer. You can pull them out. They're still alive. You can basically ask and answer any evolutionary question you can imagine, because you have the entire evolutionary series in your lab, right? Frozen available to reconstitute anytime, Jurassic Park style. So we're doing the same thing, but from multicellularity. We work with yeast, not E. coli. So it's a eukaryote, it's a fungus. And we start out with things where we actually delete the function of a single gene. And that gene's job is to allow daughter cells to separate after cell division is complete. And you get these cool fractal like tree growth forms as a result, where daughter cells remain attached to their mothers. Their mothers, every time they bud, they stick a new branch off. Each of those branches has the same sort of topology, and you get this fractal dimensionality to these branching, bushy structures, but they grow in 3 dimensions, so they're kind of spheres. They have a life cycle. They grow until they hit spatial constraints, whereby cellular packing exceeds the strength of a cell-cell connection, and they break a branch. And just like cutting a branch off a tree, if you break a cell-cell connection, that whole branch separates. There's a single-cell genetic bottleneck. The break point is a single cell, and that has daughters, granddaughters, great-granddaughters, great-great-granddaughters, etc. So the whole group coalesces its genealogy back to that single cell that broke off from the parent. And now these things are growing. They have a life cycle where they grow and divide. So if you stick a single, we call them snowflake yeast, into a tube of sugar water, basically, they'll make you 100 million by the end of growth. so they readily repopulate their environment. So they have reproduction. And as mutations arise spontaneously, randomly, they're partitioned out between groups. Groups never come back together. They're clonal. And so what you actually have is this really cool system where with very few inputs, we just broke one gene. You take a single cell organism, it forms groups. Those groups have a life cycle such that they reproduce. And it turns out that they segregate genetic variation between groups very neatly. And as a result, you can have mutations acting at the cell level, which create new traits at the group level, because you change the behavior of a cell, and that has an emergent effect on the composition and behavior of the group as a whole, even without any developmental innovation that sort of allows mutations at cell levels to create new patterns. That's just built into the system, that if you have something which grows, you can perturb a cell level behavior, and you get an emergent multicellular behavior. And natural selection sees those multicellular traits and can act upon them. We do the simplest experiment possible. We grow them in a test tube. We let them expand roughly 100-fold every day. And then we do a race to the bottom of the test tube. Actually, we pull out only 10% of the population. Let them do a race to the bottom of the test tube. Take the bottom of that, put them back into fresh media, and let them grow. So they have to grow fast, and they have to sink fast. And the main way in which they evolve to sink fast is through evolving to be bigger. Now we're doing this over the long term. So we are on transfer 1,000. Yeah, it's so simple.
yeah, evolve, but encouraging them to select themselves so you can get at them easily, which I think that's really clever.
That's absolutely key. If anybody listening ever wants to do their own evolution experiment, you need to make it something where in a few seconds you can select on a million things and pick the winners. Because if you're going to do it, like we've done versions of this experiment where you use a laser sorting flow cytometer to find individual groups and stick them into a different tube.
Yeah, that sounds hard.
It's so hard. It always breaks. Something goes wrong. You have to come in. We do our transfers 7 days a week, 365 days a year. So, the student who's doing this is tired. They celebrate. They pass their quals. They get drunk. They're hungover the next Saturday. They totally botch the experiment. I mean, these things happen. It's a guarantee. It's a statistical inevitability. Yeah. So keep it really simple. Be able to, act over 1 million of them with 10 seconds, which in our case, we can transfer, 20 populations in maybe 15 minutes, all told, all the steps. So it's really easy to do. And each population has initially maybe on the order of 1 million groups. And then as they evolve to be bigger, that number goes down because the cell number stays conserved, roughly speaking, but the group size increases dramatically. So our group population size shrinks. And so in the tubes so we have 3 different metabolic treatments in our multicellular long-term evolution experiment, which I'll just give you the little pun here. So Rich Lenski's long-term evolution experiment, the famous one, is called the LTEE, and ours is the multicellular long-term evolution experiment. So it's the MULTEE, M-U-L-T-E-E, and multi for cellular. Ha! Anyway.
Nice background. You did it.
I named it with a former student of Rich's. We were just chatting one day on Twitter, and it was good. So we have 3 major treatments. We have yeast that start out genetically identical, 1 treatment of 5 replicate populations. So They're initially exactly the same, and there's 5 tubes. So we can now look at convergent evolution in our own system. 5 tubes, we broke their mitochondria, so they can't use oxygen at all. They're just fermenting. We have 5 tubes where they can only grow by using their mitochondria. They can't ferment. So only fermentation, only oxygenic respiration. And then we have a treatment where they can first ferment and then respire the ethanol. And this was actually key because I've been working on this system for 12 years. I started it when I was finishing up my PhD, and it was a side project.
That's amazing.
And then I did a postdoc based on it, and then I got it started a lab in 2014 based on this system. So I've been working on this for a long time. And I tried a bunch of experiments where our snowflake yeast got a little bit bigger and then just plateaued. And size is really important because it's the lever for complexity. If you pull on size, you create all these side effects that are really hard for them to deal with, slower growth, diffusion limitation, and that provides opportunities for refinement, innovation, breaking fundamental trade-offs. Initially, they're evolving along a trade-off between getting bigger and growing slower. And if you can break off that trade-off, now you're actually getting things that are more complex, right? They're getting integrated. They're evolving novel behaviors that allow them to get better. So that's really, for us, like that's the philosophy. So you can pull on size as a simple lever, and you should get complexity as a way of being better while being big, right? And so it didn't work. Our early experiments just, they got a little bit bigger and then they plateaued for like a year. And the students doing this were getting really frustrated. Like, is my whole PhD going to fail? What's going on? A postdoc in my lab, Ozan Bozdog, started these different metabolic treatments with varying oxygen. And that was actually the key, because it turns out that the treatments with no oxygen, they evolved to become very large, like bigger than fruit flies. So they went from being microscopic, 100 cells, you can't see them with the naked eye, to having 1 million cells. They're 20,000 times bigger than their ancestor. And if we look at the physics of the group, we can actually look at the toughness, which is like the amount of energy it takes to break the group, scaled by volume. So the toughness of a toothpick and a 2x4 is the same. 2x4s are just stronger because they're bigger. And their toughness goes up 10,000 fold during this time. So what it turns out they do, they have this kind of cool innovation where first they make their cells longer and longer and longer. And that allows these things, which grow as a branching process, to just basically become fluffier, and they're taking up more three-dimensional space. They're not packing and jamming and fracturing. Cool. That's kind of simple. And everyone does that. Like that's what all snowflake yeast do. That's highly convergent. And there's really interesting genetic traits that allow that to happen. But then we see this change where they begin to actually entangle their cells. They begin to wrap their cells around each other. And once they start to wrap their cells around each other, that is sort of a phase transition, where as soon as some cells are entangling with other cells around them, that kind of percolates throughout the group. And now to break a branch away, it's like ripping vines off of, your bush, your fence. It's really hard, right? As soon as you pull on one thing, you're pulling on the entire entangled group. And that means that the amount of energy required to fracture these things goes up by a factor of 10,000. Which is pretty cool.
And that's just fermentation, right? These structures.
These guys are just fermenting. Right. Yeah, none of these things happen in our aerobic treatments, and they've been evolving now for about 6,000 generations, which is about 1,200 daily rounds of growth and size selection, and they basically aren't any bigger. Again, the constraint of being big on growth is just too large for those that are using oxygen. For those that are fermenting sugar, diffusion limitation is not as big of an issue. We throw a lot of sugar in their media, so it diffuses pretty rapidly, whereas oxygen gets pulled down in the liquid pretty fast. They're in a classic microbiology thing, like a shaking flask, where the top is open to the air, and you're shaking them really fast, 250 RPM. So these things are pretty tough. They're being shaken at 250 RPM 24 hours a day, right? And they have to not break apart during this time. But still, if you put like a O2 probe in the media, you see it goes from like high oxygen down to quite low oxygen after, as the population size exponentially grows up, so does the oxygen kind of mirror images that falls off. Even though it's diffusing in, it still goes down to low levels. And if you look at how far it diffuses into a group, it only goes a few cell layers thick. We can use really cool probes where we actually label the mitochondria with a charge-dependent fluorophore. And then you can actually see different colored mitochondria depending on the membrane potential of those mitochondria and if they're working or not. So you can see that basically it's only going 2 cells thick into a group. And if you have a group with 1 million cells in it, it's a pretty small proportion of the biomass that's able to get that oxygen. So small things, basically just oxygen is this sort of tyrannical constraint on size in our system, that if they have access to it and there's not a ton of it around, they never get bigger than a few hundred cells. But if you remove it, they get to be 1 million cells. And if you pump it in, they evolved actually to be bigger faster than anything else. But we never extended that experiment to long enough time periods to see macroscopic multicellularity rise. Those experiments are hard to do. We have to have a special apparatus to pump in oxygen. It tends to contaminate a lot with bacteria. And so we did it long enough to get a really nice paper, and then we stopped. We're not going to do that one.
Right? It sounds like the most intriguing part of the process.
Yeah, it's a pain. And honestly, I think from the anaerobic ones, we can still learn it.
Yeah.
You know,
can
like
cry into your Nobel Prize, I guess.
maybe. Well, it's not so absorbent, but
yeah, exactly. The gold.
So, and then I can tell you some recent results that are not yet published, but are really exciting for us. So, a graduate student in my lab, Kai Tong, has led a single-cell RNA-seq project to see if we're seeing the evolution of cellular differentiation, which is, in some sense, a holy grail of this kind of experiment. You know, you start out with something where they're multicellular, but almost in a technical sense. Yeah, there's multiple cells, but they're not organisms. They're not integrated. You can break them apart to single cells again, and they're okay, right? And over time, we expect that sort of multicellular organismality, which is a word I don't recommend saying live on the air unless you practice it.
Yeah. I'm
I've seen professors fail at that one in class. So multicellular organismality, which philosophers actually argue is sort of distinct from what an individual is. Like, it's an organism is something with integrated parts, basically. We expect that, we see that in plants and animals. If you disassociate the cells, most of the time they die, not always, right? But most of the time they do. And that's because these things have evolved to have interdependence of parts. And we expect that to evolve gradually. That's not like a binary thing. We expect that to be an outcome of refinement adaptations on being in a group and being a better group and breaking off those trade-off curves and stuff like that. So getting cell differentiation, that's a fundamental part of this. We wouldn't care about multicellularity if maximal complexity was a small clump of similar cells, right? Like, we'd be like, oh, cool, it's a biofilm, or like, oh, it's a group, but we wouldn't really be excited by it. We are excited by multicellularity because of the complexity of the organisms that have evolved, by the opportunities this creates for novel ecologies, and by the way in which these new kinds of organisms have changed our planet, right? Like, multicellular plants contain 80% of all biological carbon, and that includes all bacteria and archaea, like, by one estimate at least. Like, this is a global changing phenomenon.
So just to understand, so like, when you have the anaerobic yeast that are going purely through fermentation, and you're finding they're changing their structure to hold on to each other, is there any kind of differentiation in their shape, in the way that they're, someone is using their left hand to hold on with their right hand, or are they a perfectly interlocking system that is, I don't know, like, you've seen patterns where they just happen to all lock together, but it's the same shape again and again and again?
Yeah, it's more the latter. It's not quite as crystalline as you might expect. It's more stochastic, right? So it's more like, if you imagine what fibers look like when you're zoomed in on a piece of paper or something, where you have a bunch of interweaving, pieces. It's more like that, right? Not quite MC Escher, perfect tiling or something.
Yeah.
But so we don't really have stereoscopic integration or anything like that. But in the anaerobic lines that evolved to be large, what we did was, every 200 days of evolution, so from 0 days all the way up to 1,000, we pulled out an isolate. So they're one clonal genotype for each time point. And then we grew those up and we stripped them down to single cells in a really slick procedure where we can basically digest away the cell wall, get single cells. And then we can sequence the gene expression of each one of those cells. It's a really cool technique. It's very, very cutting edge. Everyone's proposing it in their grants and stuff these days because it's really powerful. Gene expression analysis has been around for a long time, but now you can do it at the single cell level. That allows you to see information about heterogeneity and division of labor that you couldn't see if you just took a whole organism, mushed them up, and asked what genes are being expressed. And when we do this, we see that they start out with really just one cell state, which is a growing cell state. And over time, they actually evolve 3 cell states that are very distinct in their gene expression. we always have the growing cell state, and in some sense what we think is happening that these guys are kind of almost like the stem cells. They do division, and then some of those cells that they make turn into different kinds of cells. And then we have a cell state that is expressed primarily, so I have to be a little careful because this is all work in progress, and this is stuff that has not been published yet, but is I'm pretty confident about. The gene expression analysis is done and it looks really good. So I'm very confident in that. But exactly labeling which cells are doing what and why they're doing it, that's what we're working on right now. So with those caveats aside, I'll tell you what I think is happening. So we have a subset of the cells that are actually expressing a lot more cell wall material. These cells, if you look at the genes that they're turning up, these specific cell wall biogenesis genes, and you label those products that they're making, you see that it's the hub. So we have sort of this network of cells, and that means that you have hub cells that are old and have a bunch of branches attached to them, and they're highly connected. And that's just a consequence of older cells have had more time to grow and divide and have more daughters, and they have these big branches of cells that are sort of tugging on them. And our biophysical models, we've done some really nice 3D biophysical models with Peter Juncker at Georgia Tech. They suggest that's often where the breakage occurs in our groups. So if you want to make the groups stronger and bigger, you should reinforce those specific hub cells and make them stronger. And what we're seeing is in the ancestor, that's not happening. But by 200 transfers, 400 transfers, all of these guys downstream, they're cranking up cell wall biogenesis, and it's being expressed in a subset of those cells, the ones that need it, that are at that hub. So that's pretty cool because you're kind of seeing this biophysical differentiation.
And so is that sort of similar, like epigenetic, like like you've got the cell realizes the conditions that it's in and expresses different genes depending on its situation, whether it's at the outside or whether it's one of the structural components.
Right. And so there's different ways in which you could get there as a cell. We don't know what the regulation is. Is it there's two basic things we're investigating. One is just that it's an age-dependent thing. One of the neat things about our system is that if you're old, you're a hub cell. And yeast are actually very good at knowing how old they are. They do all sorts of things on a clock. And so there may just be its internal clock ticker, right? And so what we're doing right now is we're actually turning these things that evolved to be multicellular. We're genetically engineering them back to unicellularity. And then we can look at their behaviors when they're not in a group and ask, are they still doing it, or are they no longer doing the same behavior? It's a pretty cool, pretty powerful technique.
And so how, I guess, when I think about animals like me, I've got hair cells, I've got skin cells, I've got, liver cells, et cetera. And you've got this differentiation that is just continued and continued and continued. And so is the hope that you're starting to see the beginnings of this differentiation in your yeast. And if you run the experiment forward, you will get to some point where it's clearly making outer membranes, inner structural elements, et cetera.
That's exactly right. Yeah, so one of the neat things about what we're doing is it's exploration. Like I do not know what's going to happen. And if I had hypotheses, they're only as good as my prior knowledge. That's actually one of the things that is wonderful about our approach, is that in some sense it's hypothesis-driven science, but in another sense it's not. It's exploration. And hypotheses are great for finding a mechanism, for trying to get at the sort of nitty-gritty of why something does something else. But if you're in a space where you don't understand it very well, hypotheses can be a crutch, because you can only hypothesize what you already understand, or you at least think you understand. And so exploration is slightly different. We are prospectively exploring how complexity arises in real time, which will allow us to generate additional hypotheses that we can rigorously test. But there's so much that we don't know about multicellularity that we are, I think, better served by doing exploration rather than taking what we already know and trying to see if it's rigorously test those hypotheses in model systems or mathematical models.
It must be exciting to see these kinds of things starting to develop, but also frustrating in that it still takes so long. You wish you could just go forward. You've been doing it for 12 years. Imagine another 20 years. Where would we be?
Yeah.
But every day you have to come in and you have to feed the yeast and you have to collect the survivors and you got to go. There's all these.
And you're totally right. And even once you have this super exciting result, it's usually 2 to 5 years before you can publish it, because you have to just make you have to really understand it, right? So we try to do a really holistic style of science where we do the evolution, we do the genetics to identify the things that have changed. We use the genetics to identify genetic mechanism. We integrate this with fundamental biophysics because you cannot understand multicellularity without understanding its biophysical context. It's so deeply embedded to how and why multicellularity evolves. And we try and integrate all this with broader mathematical models that try and extrapolate this beyond a simple laboratory model system. So our papers they're multi-year efforts from a team from a big team of scientists with different skills. And that's really rewarding because you can do the kind of science that I frankly always wanted to do. But it's only made possible by, by strong funding, by long grants. 5-year grants are really nice. By being able to have people that are willing to invest 5 years in a project and just really go after it day after day and, tie up every loose end. And while at the same time, cool new stuff is coming down the pipe. And you're like, oh, I want to work on the shiny new thing. No, we got to get the old stuff out.
Right. Don't forget about the yeast. Feed the yeast.
Yeah, exactly.
Yeah. Mean, it is interesting that there's this division in the astronomical fields, and I'm sure it's the same in yours, that there's the theorists and then there are the experimenters. And I tend to favor the experimenters just because there's something just so joyful about just going out, as you say, it's exploration. You kind of go out into the unknown. You don't know what you're going to find. What'll happen if we just keep feeding yeast and keep collecting the heaviest ones and just let that run for tens of thousands of generations? Let's find out.
And already it's changed our priors. Considerably. So when I started this out, nobody was doing experimental evolution of multicellularity. It was frankly sort of a ludicrous idea because the thinking was, well, you're never going to make an animal. It's too hard. It takes too many steps. We don't even know what to start with and how to select for it. And, the thinking was that multicellularity was rare and very difficult. And that, in fact, we can get into this, but there's this general thinking in biology and probably in astronomy as well, that things that are rare indicates that they're difficult. And in biology, that's not always the case. You can have things that are rare because they came first and filled the niche and suppressed further innovation into that space. Or you can have, you know what I mean? And so you can have things that are common, even though they're extraordinarily difficult, because there's an advantage and evolution has many high dimensional ways of getting to different places, to the same place, given many generations of selection.
So I'd like to shift gears just to talk about about your work in astrobiology. So what projects have you been involved in astrobiology?
That's a good question. Honestly, the main thing is our yeast system. I would kind of classify this as both evolution and astrobiology, in the sense that if your question is what's the probability that we'll find complex life on another planet, then multicellularity is one of those major transitions in evolution that you need to have good priors on its difficulty, right? If you think it's rare and very difficult, then you'll say, cool, well, then the probability of getting life is this, the probability of getting cells is that. And and if we have a bunch of planets with cellular life, the probability of having multicellular life is, maybe 1 in 1,000. And then what about intelligent multicellular life? Oh, there's another 1 in 1,000. And then the chance that they're around when we're looking, well, there's another 1 in 1,000. Okay, we're not going to find them. So like filling in those priors, that's in one sense what I think a very important job of the field of multicellularity research. We're primarily coming from an evolution stance. Like I was trained in EEB, but in astrobiology, I think it's really helpful to try to figure out what's the probability of this, what are the big constraints, are there certain geochemical conditions under which this is favored. For example, is how important is oxygen? Is no oxygen a death knell? Is high oxygen necessary? Those are all really open questions, and they're very difficult to answer for anything outside of Earth. Well, and so we can do some stuff here.
Yeah. I mean, the traditional, when you do the traditional argument, say for like the Fermi Paradox, is that the universe is big and that it's old, and the Earth formed billions of years after the formation of the universe, that weirdly life arose fairly rapidly into the Earth's formation.
Super fast,
yeah, like literally as quickly as it could possibly do so. And yet multicellular life arrived later on. Seemed like it was a more difficult way to form life, and that the reason we don't see life out there in the universe is that it's entirely possible that multicellular life is something that only arose on Earth and has never been figured out anywhere else. And this is, the rare Earth hypothesis that but and it feels to me like you're kind of destroying that theory.
Yes and no. I mean, at least yes, in the sense that if the argument you made, I do think we are destroying that argument. But you can make a secondary version of that argument that's not destroyed, which is multicellularity per se is pretty easy. Like, I mean, I haven't really talked about all the other stuff that we've done in this area, but like we can kind of show that the conditions required to get a group of cells, to have that group of cells become Darwinian, such that it replicates, has heredity, selection acts upon it, and it adapts at the multicellular level. That's trivially easy. There's probably not a cellular lineage in the world that, given time, we couldn't make it do that. Like, I think that's really, really fundamental. Like, if you have replicators, you can get a group of replicators that becomes its own replicator, and that's not hard. In fact, in a sense, that's what cells do too, but just bounding it in a membrane rather than sticking a bunch of membrane-bound things together. Like, you see this happening in biology all the time. You have this hierarchically, nested doll of replicators within replicators within replicators, and that's really easy. But if you're talking about intelligent life, there's a number of bottlenecks in there that aren't really just about multicellularity, but are about a specific kind of multicellularity. And there's a bunch of singularities that are pretty difficult. So for example, so cellular life, in some sense, the hardest challenges in all of biology are getting a bacteria or an archaea, getting a cell, right? Like coupling
like the abiogenesis in the first place.
That's hard. Like, I think it's in some sense the fact that it happens so fast, the fact that I actually think that Eric Smith and what's his name? He was just on Mindscape, gave a great interview. I'm forgetting his name. Sorry.
What's the concept? What's the theory? I might be able to help you.
Origin of life.
Right. So like panspermia versus.
Yeah, I mean, so there's biochemists and chemists that are working on origin of life, and I think there's convergence in the field that what you need is essentially primitive metabolism that's primarily driven by planetary disequilibria, right? Primarily, like you have a giant battery of a planet. If you have a planet with a ferrous core and a water surface, then you're just going to have electrons fluxing from the core into space via UV breaking apart water, and then electrons from oceanic spreading or whatever, like basically going into that oxygen and flying off into space when hydrogen is liberated and fluxed out off the planet. And so you have this like just constant electric battery of Earth, which allows for disequilibria to exist. And if those energetic disequilibria can be endogenized and accelerated by chemistry, then you kind of have seeded this planetary incubator for chemical evolution.
Right.
And the difficult thing is like you have the metabolism for free. You get simple carbon chain metabolism like Fischer-Tropsch reactions occurring on minerals in all sorts of aquatic environments for free. And all you really have to do is get a TCA cycle, a Krebs cycle, run it backwards, and you can fix carbon and make all the molecules that we ever really need in biology. And you just kind of have to couple that metabolism to a Darwinian mechanism that accelerates it, and then you're off to the races because it can happen fast. And I'm actually kind of partial to that argument. So what are the singularities that are difficult with getting to complex life? The big ones are, so cells are huge and difficult, but that apparently happens fast. Once you have that, eukaryotic cells are seemingly important, right? There's no bacterial lineages that have evolved anything remotely complex in the multicellular space. You're talking about chains of cells, or at the very best, these balls of nostoc cyanobacteria that look like those balls you throw in your dryer to keep things from clumping up. They're about that size. They occur in really cold lakes, but they're just spherical balls of cyanobacteria. They're not that exciting. Eukaryotes, which are these cells that are essentially, they have this symbiosis with JV mitochondria. I actually don't personally think the mitochondria is game-changing in that many ways, but what eukaryotes have that is game-changing is a really complex architecture to the cell. They have a lot of flexibility and dynamicism in the way the cell can move. They have actin cytoskeletal networks. They have all this subcellular compartmentalization. And it's relatively easy there's a lot of possible states that a eukaryotic cell can exist in. They can be an amoeba, they can be flagellated, they can be hard. I mean, they can do everything. All the different kinds of cellular differentiation you see in plants and animals, like the eukaryote could probably do some version of that, right? Like these things aren't really new, they're just repurposed.
Right.
So eukaryotic cells have all this behavioral repertoire that prokaryotes just don't really have. Prokaryotes are primarily super crazy and complex across lineages, but eukaryotes, a single genotype can be complex, right? And so that's the main advantage that eukaryotes have. Eukaryogenesis just happened once. It's probably pretty hard to do, in my opinion. It took a while and it required a pretty wholesale reworking of the way in which these cells work. You have to have a nucleus, you have to have all these other compartments. That's hard. And that took a long time. So that's one sort of singularity that's a bottleneck. The next one is animals. Like, we don't think of plants as being intelligent, right? You can have all the cool macroalgae, kelps, plants, Volvox, there's really cool algae out there from a multicellularity perspective, but they're not going to be making radio antennas. You need something which is a heterotroph, which can have, cognition, neural networks, brains, right? So first of all, you need to evolve something like animals. Animals are their own singularity. The holozones, so animals, we call them the metazones. That's the clade that is synonymous with animals. And the holozones are like kind of one hierarchical level higher, and those include these other quasi-single-celled, quasi-multicelled oceanic critters, Philisterians, ichthyosporians, quantiflagellates, and they have all these really interesting cellular behaviors that you see in animals. And in fact, almost all the genes that we have for multicellular development, these things that aren't animals, they have them. They use them for different things. Animals seem to have just repurposed them for multicellularity. But animals only evolved once, and to some extent, the conditions were probably, had to be kind of right for that to happen. You probably needed a fair amount of oxygen, right? Especially for things like bilaterians. We don't think about this, but non-bilaterian animals really just have one or two cell layers thick. Jellyfish are like one cell layer thick with jelly on the inside. Bilaterians have multiple cell layers thick, and you're probably not going to get that without oxygen and without circulatory systems and oxygen carriers. Static oxygen binding molecules like globins, and then mobile globins, especially, that you can move around. Once you have gills, you can get really big, right? Because you can decouple size from diffusion. And that's hard. Getting animals is hard. Getting animals probably require the right genetic composition of the unicellular ancestor. Like, you probably aren't going to get animals. You're not going to get animals from an algae. You're not going to get animals from a fungus, even though fungi are actually fairly closely related to animals. You need something with like holozoan-like behavior, where you have cells that can be amoeba, cells that can have flagella, things that are already heterotrophic and are not relying on photosynthesis for generating energy. Because if you do that, you're not going to make an animal. You're going to make something which looks like a plant. You need to set the stage for something which has the genetic repertoire of animals, but is not yet an animal. And how hard is that? I don't know, but my guess is that's pretty hard, much harder than multicellularity writ large. And finally, sorry.
Yeah, no, yeah,
I got one more.
Finally. Yeah.
I got one more. Once you have animals, it's not just enough to have a jellyfish. You need animals with brains. You need animals with neurons. Neurons, interestingly, evolved twice, we think, within animals. Once in comb jellies, and once in mussels too, and once within the cnidarians and bilaterians, which is kind of astonishing, honestly, that like comb jellies, right? These things which have like luminescent patterns as they wave their cilia. They have different genes for neural transmission and muscles. And if you like use any of the neurosystem modulating drugs that we have, like that work on all animals, it doesn't work on them because they're not using the same genes. It's really cool.
That's amazing.
So
I had no idea.
So once you have an animal, basic animal, it's probably not that hard to make a neuron. In fact, we have it convergently evolving, right? And neurons are just bootstrapped up bioelectric cells. All cells can communicate with other cells bioelectrically. Every cell uses an electron gradient to generate energy. And so being able to tweak other cells' electron gradients is pretty easy with, membrane-bound proteins that can be dynamically regulated based on voltage. And so basically the building blocks for cells to communicate bioelectrically is just embedded in the system of how cells work. And neurons are just super specialized versions of regular cells, right? And so then once you have a nervous system, how difficult is it to evolve a brain and have intelligence, right? Intelligence in the sense of being able to, within generation, plastically fit your environment and do all sorts of complicated things. That's pretty hard too, although brains appear to evolve once in bilaterians, but big brains evolved twice or multiple times within within mammals even, but also you see like octopi and squid evolving big brains pretty independently of the rest of animals. Like, they're sharing a common recent ancestor with a mollusk, with a clam. And so, an octopus brain is a pretty remarkable achievement for a clam. So I actually think that that's possible to do, but, difficult. And then once you have a brain, getting a brain sufficiently large that you can have civilization scale cultural evolution take place, which is what you need if you're going to generate the resources required to have technology and be detectable across the universe. That's pretty hard too, right? You had to have primate brains, which have a different fundamental scaling law between brain size and neuron density. Most brains actually begin to plateau out. You make bigger brains, but you're not packing the neurons as dense. But bird brains and primate brains break that trend. And basically, we just have a scaled up chimp, monkey brain. Our brain has the same density as a capuchin, but it's just really big. And so it's relatively easy to just pull on that lever of making our brains bigger, having us evolve that in a social context, allowing for communication. Once you have communication and societal level scales of stores of information being preserved, you can have the kinds of technological development that you could detect across the universe. But there's a whole lot of singularities baked into the story I just told you, and really not any of them rely on multicellularity other than in the sort of most proximate sense.
Right. And if you had to pick one of those, would it be eukaryote?
As the most difficult?
Yeah.
I'd say animal.
So
if
animal.
I had to pick, I'd say animal. Yeah.
So a sort of Swiss Army knife of a cell seems possible.
Yes.
A single cell, but that's really under a lot of evolutionary pressure to figure out how to be able to be adaptable in a lot of places. But for them to come together and differentiate and form an animal feels like a pretty big jump.
I think mobile heterotrophy with muscular motility dictated by a nervous system can like that type of behavior. It's maybe not the biggest jump, but it just seems so dependent on having the right system, having the right cell biology, and having the right Earth scale situation where you need the unicellular ancestor with that repertoire of behaviors. I think you do need actually a lot of oxygen to get something which is animal-like, because it's heterotrophic. It's going to be burning a lot of O2. Because it's big, it's going to need fairly high O2, even if it evolves things like gills. And maybe actually like you could survive going back lower O2 once you had a high O2 intermediary where you could actually overcome because they don't start out with gills. They start out needing diffusion. To me, that just feels more dependent on cellular, like biological and Earth priors that kind of Goldilocks have to come together. Whereas eukaryogenesis, to me, I think there's more opportunities for that. You can chill in a deep sea hydrothermal vent and be kind of ignorant of what the rest of the world is doing and become a eukaryote. Like that's kind of what we think has happened, right? Like the things that are not eukaryotes, but are the most eukaryote-like, are these Asgard archaea, named after the Norse gods, and the Loki archaeota and the Heimdall archaeota, which are fun to say, are these archaea which are found in these deep sea hydrothermal vents. That they are very much archaea. They don't have mitochondria, but they have all these genes that we thought once were only found in eukaryotes. They have complex cells. They're interacting with bacteria symbiotically in their environment in ways that like really looks like a precursor to domesticating mitochondria. They're doing chemical metabolism with like bacteria, and they have these long elongated arms that they're interacting with them. It's one of the coolest discoveries in the last 5 years, honestly, in my opinion.
You're doing an experiment to essentially to try to make yeast into a eukaryote.
Well, they're already a eukaryote. To
more differentiate then is the goal to make that transition to animal, or can you envision a different experiment, same thing, thousands of generations, to try to go from pre-animal to animal?
Yeah, we're not going to get animals because they have a fundamentally different ecology. Our guys aren't modal. They're not really tracking down prey, like that kind of thing. What I want to see, I'll answer the second question next, but what I want to see with our system is like, I think snowflakes are their own thing. They're their own organism, and they're going to evolve bodies and division of labor. I didn't even tell you about the third kind of cell type, but it doesn't matter. but they're going to evolve bodies and division of labor in ways that make sense for them based on their mode of growth, based on the selection that we're applying to them, right? Like based on the toolkit that's available from their unicellular ancestor, which is, broadly speaking, that's true for every single thing involving multicellularity. That's the way in which it works. And so, I want to see how far we can push them. We're already getting to the point where reverting them to unicellularity genetically is very difficult, if not impossible. I want to get to the point where, I would like to know at a certain point, if we break them back to unicells, do they die? Are they like animals in that sense, that like single cells just perish? They're not able to persist? How much developmental innovation can we get? How complicated and how organized and how precise and how robust can their bodies become, right? How many different cell types can we get? How much transcriptional regulation can we see? Like, these are all really open questions that I'm delighted they're evolving as quickly as they are, frankly. Like, I know it's slow, but it's actually faster than I expected. Like, didn't expect to see cell type differentiation so soon, and yet I think it's unambiguously happening. So the second question is, how could we get something more like animals? And I think what you want to do there is take something which is basically from that holozoan group. So in fact, we started a project doing this and COVID killed it, sadly. So yeah, you want to take like a quantiflagellate. Nicole King is, one of the world leaders, I'd say the world leader in quantiflagellate animal relation multicellularity. Quantiflagellates are these cool creatures that live in the ocean. They share a common ancestor with animals like 1.1 billion years ago. They look like a sponge. In fact, the individual cells in a quantiflagellate are almost identical physiologically to the cells in a sponge. They have a little collar, they have a flagella, they whip the water around them, they suck the water through, it gets caught on that collar, which has mucus on it, and they eat bacteria that are being filtered through them. So they're basically doing at the single cell level or at the small multicellular group level what a sponge is extremely good at doing, pulling water through it and filtering out the bacteria and eating it. So Kai Tong, this great PhD student in my lab, started an experimental evolution project with quantiflagellates where we were basically evolving them in two different ways: size selection, like our snowflake yeast. What happens if we pull on the lever to get bigger? How do they do it? Can they, what happens? And then also one where you're trying to make them adhere to a substrate and become more sponge-like. So, stick like a little blob sticking to a glass slide and filtering water through them. And then Kai went back to China in fall of 2019 December of 2019 to renew his visa, and COVID hit and he was locked out of the country for a year and a half. Yeah. And it destroyed that project.
But that was how you would go about it, you think?
I would either use quantiflagellates, or there's another researcher in Switzerland, Omaya Duden, who works with a different holozoan, the ichthyosporians, and he actually has a system which has proto-embryos. Like, it forms a sphere and it has like polarity, and then it just like sporulates and makes, so it forms a sphere of cells that has polarity and it looks like an embryo, but then it just explodes with a whole bunch of spores. So it looks like an animal embryo until it explodes with spores. It's really, really, really cool. And he's really keen on doing some experimental evolution as well. And I think we're going to collaborate on that project as well.
It's interesting. I mean, it seems like going after what appear to be the rate limiting steps and then trying to force the evolution in the lab will give a lot of information about how difficult it might actually be. I've got one, we're sort of running a little over time. How's your time? Are you okay? Okay.
I'm fine. Yeah.
Okay. So I want to talk about this idea of a shadow biosphere. And this has been proposed by many people, that there are life forms that that are existing within our environment, but we don't have we're not looking for them, and so we just never notice them. Do you think there's any possibility of a shadow biosphere?
Can you tell me more? I have my own thoughts on this, but I want to get a little more insight on what you're thinking.
Well, I don't think anything.
Okay.
I mean, I think that, it doesn't seem, me personally, my job is to ask the questions. My opinion is that that we all use the same matter, energy, and there would be waste produced in certain ways that would be obvious, and we wouldn't know where that came from. When you think about a seabird colony that's there's tons and tons of bird
Mm-hmm.
poop everywhere. Where did
Mm-hmm.
the bird poop come from? We would see
Right.
giant deposits of,
It's like a dark matter question.
yeah, we would see exactly. We'd see giant deposits of stuff and go,
Yeah.
where did that come from? There's no biological process that's doing it,
Yeah.
and yet
Yeah.
it just keeps appearing out of nowhere. Must be the shadow biosphere. And so I think if it was, I guess, if there was enough out there, then we would see the evidence, macro evidence that would be obvious and begin the place for research. That's my feeling.
So I got two thoughts on this, and one is very real, and one is currently sci-fi. All right, so the very real one is that there is a shadow biosphere of a sort, in that we do see it if we look for it, but we typically haven't looked very much, and that is the deep Earth biosphere. Basically, no matter how far down you go in Earth, you pull samples, you find living things. It's ridiculous. You find them inside rock. You can dig up mud that has been in the seafloor for 1,000,000 years and separated from the surface, and it's full of living bacteria, and they have highly reduced genomes, and it looks like they've just been circling the same carbon over and over and over, and just fragmenting their genomes until they become highly specialized on different, smaller and smaller pools of food that's available.
Right.
You go
They're all
down
sharing
to,
the same carbon atom, just
yeah,
with a little
yeah,
bit of heat.
and they have like,
Yeah.
you can estimate their doubling time, and it's like 10,000 years.
That's amazing.
It's ridiculous, right? You go down to gold mines a mile deep. You tap the water, and there's like a single species of bacteria that's eating hydrogen that's being created by radioactive decay, and like that's really, I think that's really cool. Like the fact, I mean, it basically means that outside of like the sun essentially turning us into Venus, like there's no way to sterilize this planet.
But we
So
share a common
thoroughly
ancestor with
suffused.
that.
We do.
Right.
Yep.
So
Yep.
something that we don't share a common ancestor that is still existent on the Earth.
Okay. I don't know of any evidence for that, but here's the sci-fi thing, and what made me really think about this seriously for the first time was a paper 2-3 years ago where somebody made a mirror image DNA synthase. And so basically it copies DNA, but it makes a copy that has the opposite chirality of standard DNA. So the really interesting thing about this is that all the enzymes that work on modern DNA, standard DNA, it doesn't work on it, right? You can take this stuff, you can put it in pond water for a month. They did this, and it's completely intact after a month. Whereas you take regular DNA and it's gone in like an hour because everything wants to eat it.
Yeah.
And I mean, I could be wrong. I'm not a biochemist, but the thing that got me thinking about this as oh my God, this could be a phase transition where humans inexorably change the way in which life on Earth works, like forever, is if you were to make a mirror image ribosome and a mirror image cell where everything was the opposite chirality. You could make something which basically wasn't in ecological competition, really, maybe for like metals or something, but it wasn't really an ecological competition with the standard biosphere, wasn't impacted by toxins from the standard biosphere. There's a fair amount of automatic chiral switching that just happens spontaneously, spontaneous racemization. So there's kind of crumbs. I think there's crumbs on crumbs up from the feast of the standard biosphere that's something which was mirror world biosphere could actually consume. And so if somebody were to make a mirror world pelagibacter or a mirror world, whatever environmental bug and release it, it seems plausible to me that you'd have something which would then have a completely open ecological niche where there's all these little metabolic crumbs trickling down through spontaneous racemization from this huge biosphere, and they can essentially evolve and occupy different niches with very little competition. Viruses wouldn't impact them, right? Antibiotics wouldn't impact them. They'd be competing for space, they'd be competing for light, they'd be competing for metals, but nothing else. And wouldn't that be a trip? Once you open that bag, I don't think you're never going back, right?
Yeah, that's really interesting. I mean, it's sort of similar to this idea of people talking about like aliens and just like, what if the aliens were so different that we couldn't even comprehend them? And that's absolutely possible, but we share a universe. We share radiation from the stars. We share the various chemistry that exists in our.
That's right.
But if they, for example, were based on opposite chirality life, then there would also be vast swaths that we wouldn't be competing with them about, and maybe wouldn't recognize their their processes and wouldn't be able to detect them. If you were measuring the chemicals in the atmosphere on some exoplanet, and you wouldn't be able to detect the outgassing that you would be expecting, then it would sort of throw the whole search into doubt. So it's very fascinating. It's a very fascinating idea.
Isn't it a cool idea? And the proof of
Mhm.
principle of making this in the lab, the mirror image DNA synthase, and making mirror image DNA. It's like, it's not just sci-fi. Like this is within the next 100 years, someone could probably do this.
Wow, it's really fascinating.
I mean, a ribosome is much harder than a DNA synthase, but it's not that hard.
Yeah. Well, William, it's been absolutely fascinating talking to you.
Yeah.
If people want to keep an eye on your research and hear, the latest generation of the yeast and what it's up to, what's the best place to do that?
Twitter, for sure. So whenever we post all of our papers as preprints, because it usually takes a year or more to get it finally published after all the revisions you have to do, et cetera. So I publish all of I push all of our preprints to Twitter. You can just see me talking about random biology stuff there too.
Absolutely fascinating.
Yes.
And what's your
Thanks
Twitter
for having
handle?
me on. WC_Ratcliff.
Perfect. All right. Well, William, please let me know when one of your yeasts sort of crawls out of the petri dish. And
if I'm still here after that event, then I'll let you know.
That sounds great. All right. Well, thank you so much.
All right. Thanks, Fraser. Take care.